For many of us, it might be our first actual science seminar of the year, which is pretty exciting for us science nerds to be able to get together and hear a seminar and have some good questions. And before we start, I'd like to acknowledge the traditional owners on the land in which we meet and pay our respects to their elders, past, present, and emerging. Just for those folks here in the room as well, we're going to do some housekeeping. Drew just told me we should do some housekeeping. If there's an alarm, please exit in an orderly fashion. The doors are where you came in, and there's toilets around the back. At the end of our meeting today, we're going to take some questions from the Zoom world, and we're also going to take questions from the audience here as well. We'll try to make sure everyone can hear each other. And we're going to have, for those people here, we're going to have some beer, wine, and soft drink, and some pizza as well to celebrate science. We've been running EnviSMART now for 2 years. Actually, Drew's played a really pivotal role in helping us set this up. And we set it up when COVID started, and really it was about trying to maintain a sense of community for the environmental science professionals, educators, consultants throughout the country. And so we've been doing this every month now for 2 years now, which has been a lot of fun. And we can only do that with the support of partners. And I'd like to acknowledge those partners, which are Agilent and Eurofins, a couple of Eurofins folks over here today. Also the Australian Contaminated Lands Consultants Association, Water Research Australia, and CTAC Asia, Australasia, and the president, Kath Hassel, is here today. Thank you very much for your support, Kath. And also Trajan, PM Separations, and Senversa. Well, I'm going to get out of the way, and we're going to introduce you to your keynote speaker today, who is I have the great pleasure to introduce. Drew has been working on his PhD for about 3-4 years now, so he's about to submit, which is really exciting. And I don't actually have your bio. I'm going to see if I remember it. Drew did his undergrad at the University of Western Sydney. After that, he worked and realised he didn't like it, and then came and did a master's in environmental science at RMIT University, where we first met. And then he decided he enjoyed it and wanted to continue doing a PhD. And midway through that, unfortunately for him, I decided I wanted to move universities, and Drew came too, which is great. And so he's been a really pivotal member helping me set up the lab, which is called the Australian Laboratory for Emerging Contaminants. So without further ado, I'm going to welcome Drew to the stage.
Thanks everyone. Welcome. Thanks for coming to see probably my last presentation as a PhD candidate, fingers crossed. So I'm going to be talking about, like Brad said, the work that I've completed over the last couple of years. Some of this information you might have seen at previous conferences, but there's a couple of new things that I'd really like to share with you today. So there's a lot to do. Let's get started. So I'm speaking to you today from Wurundjeri lands, and I'd like to also acknowledge the traditional owners of the lands, for those of you at home as well. So first of all, here's a list of my research aims. These were actually developed in collaboration with Melbourne Water and Water Research Australia. And while this work is really important to the water industry, we are also breaking ground in terms of research into PFAS in Australia. So when I first started with Brad, there were only a few research labs in the country that were working on PFAS contamination, and research in general was still very much in its infancy. So each of the projects that we selected were developed to really answer these questions for the first time in the Australian context. And often they hadn't been described internationally either. So for point 1 here, I conducted the most high-resolution study of PFAS concentrations from a wastewater treatment plant ever done. Point 2, we looked for contamination in a local Melbourne lake, and quite serendipitously, we were able to describe the bioaccumulation of legacy and novel PFAS for the first time. So in that study, we also discovered a novel sampling technique that may be really important for future biomonitoring studies. So today I'll be discussing all of these findings, focusing on the first 4 chapters of my thesis. So I'd like to add here that I won't really discuss the toxicological impacts of PFAS. That'd be another 40-minute seminar by itself. Suffice to say, the exposure to PFAS is not good, and the aim is to mitigate that exposure where possible. So I've mentioned PFAS a couple of times now, and some of you will be familiar with these compounds, but let me take a few minutes just to bring everyone up to speed. So here's the general chemical structure of PFAS, right? A fluorocarbon chain on the left in blue. Some PFAS have a normal hydrocarbon chain in the middle, so that's a fluorotelmer in dark blue. And then all the ones that we'll be talking about today have a functional group, which is in green. So this is also how PFAS are named, firstly by the number of carbon atoms in the chain, and then for the functional compound at the end. So taking PFOA and PFOS, for example, perfluoro, meaning all carbons are fluorinated, octane, meaning 8 carbons, and either oic acid or sulfonic acid for the functional groups. Now, we know PFAS are extremely hard to break down, travel long distances in the atmosphere and oceans, and can be relatively toxic at low concentrations. So most of the usual suspects like PFOS and PFOA here have been routinely monitored over the last 20 years and were since regulated. And then a whole new crop of compounds were developed to get around some of those regulations. In fact, now there's over 4,700 individual PFAS compounds recognized by the CAS registry. Many of the ones that we now detect in the environment come from around 40 classes or 40 functional groups. Within each of these classes, there'll be compounds with multiple chain lengths that affect how they move around in the environment. So these replacement compounds are still being used in products like firefighting foam, and I'll call this AFFF, and in products that you use at home like Gore-Tex and Scotchgard. One of these compounds is called PFECHS, which is associated mainly with aviation grade hydraulic fluid. So it's comprised of 8 completely fluorinated carbon rings and a sulfonate group, just like PFOS, except it's a cyclic compound. So it's used as an additive in aviation grade hydraulic fluid because it's resistant to fire, corrosion, and evaporation. And many aircrafts today still use PFECHS as an additive in the hydraulic fluid. So the acute and chronic toxicological impacts of newly detected compounds like this in humans and wildlife are still not really known. But part of what we do is to understand the sources, transport, and bioaccumulation to help effectively quantify the risk to the environment. So you might be asking yourself, well, how have these compounds been detected in all kinds of biomes around the world, from Antarctica to the Arctic? Well, where there's no factories, Well, here's an example of how these compounds are transported. You can start with a compound called 8,2-FTOH, which has an alcohol functional group. Because it's an alcohol, it can volatilize and enter the atmosphere. During its travel around the world, it may oxidize to another form called 8,2-FTCA. So now it's changed from an alcohol to a carboxylic acid, and it's more soluble, so it will fall out in the rain and snow and enter our rivers and oceans. From there, it can be oxidized again to its terminal product called PFNA, and this can travel around the world's oceans and wildlife for quite literally hundreds of years. Notice how all these reactions only occur at the end of the compound where there's no fluorine. This is what makes all PFAS so persistent in the environment. They're called forever compounds because there's no known natural process that will split the bond between a carbon and fluorine atom. They'll stay like this and not react for a really long time. Let's zoom in now to a more local area and see how PFAS might move around a city. So on the left, you have the industrial sites and firefighting training facilities, which are the primary sources of PFAS. Remember, these aren't naturally occurring compounds, so we have to produce these ourselves. They can enter the environment on purpose by spraying firefighting foam or AFFF, or dumping the waste legally down the drain. There's also accidental spills sometimes that get washed into local waterways or end up in the groundwater. The real problem is when they get to a sewage treatment plant. So wastewater treatment plants and sewage treatment plants are designed to remove human waste, mainly nitrogen and phosphorus, so the water can be discharged safely back into the environment, right? They are not designed to remove PFAS. So most of the time they're emitted directly from the treatment plant unaffected. So what happens if a treatment plant uses some of the recycled materials like water or biosolids for agriculture? Now the chemical is being applied directly to the food that we consume. Now that's why nearly every human on the planet has PFAS in their bodies right now, but usually a concentration so low that it's probably not going to cause any harm. So my whole project has centered around the discharge of PFAS from these treatment plants and how they can affect local waterways and the birds that live in them. So we're going to look at why we use birds as biomonitors first. So birds are really useful biomonitors for chemical pollution in the environment because they typically have really large foraging areas and they can feed over multiple trophic levels. For example, here's a map showing the feeding areas for a marine seabird off the coast of Morocco and Portugal. So the black dots are where the birds are nesting, and the shaded areas are where the birds go to collect food. These birds will travel hundreds of kilometers a day to fish, and they also eat anything from snails, which are low on the trophic order, to crabs, squid, and octopus, which are much higher in the trophic food chain. When we study these birds, we can use that information to monitor pollution, but also things like the levels of fish stocks and climate change by looking at the health and success of a particular individual. But more often, we use the entire population to get an idea of what's happening on a very large scale. Declines in bird populations are often attributed to depletion of food stocks, as overfishing by humans has left very little food for the birds to eat. For chemical pollution, we have been using birds for a long time. Think the canary in the coal mine. We use birds to tell us when there's low levels of oxygen or toxic gases in mines. More recently, we can use really advanced techniques to quantify these effects. So when you graph the trophic level and the concentration of a chemical across an entire ecosystem, you can see that organisms lower on the food chain have lower concentrations. And as each prey is eaten by its respective predator, the concentration increases in the body until those animals at the top of the food chain, in this case, birds, the levels are extremely high. So the scale on the y-axis is a logarithmic scale, which means from each trophic level, say from fish to birds, there's an increase in concentration by a factor of 10. So another important thing to point out is that different types of birds will accumulate PFAS at different rates. So the rate of accumulation has been found to strongly rely on the bird's trophic level, but also if they feed in marine environments compared to terrestrial environments. The level of nitrogen isotopes in birds can be used to see what trophic level the birds are feeding at. And a higher number here means that the organism is eating from a higher trophic level. And then on the x-axis, you can see the levels of carbon isotopes can be used to tell if the birds eat at sea or on land. So here's the diversity of a different species and colonies of gulls from one region. So the birds on the left are marine feeders and the birds on the right are terrestrial feeders. But we can see that the level of contamination increases as a function of both of these factors. So high trophic level birds tend to have high concentrations, as we said, but also marine feeders tend to have higher levels compared to terrestrial feeders. So here are some of the accumulation factors for different types of birds that have been studied in the wild. We have marine seabirds, a freshwater duck, and an eagle, and they will accumulate PFAS between 50 to 40 times the level of what's in their prey. From 89 biomonitoring studies conducted over the last 20 years, many have focused on birds from the Northern Hemisphere. And of these, most of the birds studied were from the Arctic region. And that's why Canada and Norway both have the most number of studies. From all these studies, PFAS has been detected in nearly every bird sample. So here's an idea of the range of concentrations that you would typically see from these areas. So you have a 3M facility where the highest concentrations ever found in birds was roughly around 190,000 nanograms per gram. In the North American Great Lakes, you see concentrations up to around 400 nanograms per gram. So the Great Lakes are actually heavily impacted by nearby fluorochemical plants, airports, and wastewater treatment plants, but they're quite large, so it's very dilute. Still in the Arctic, you can find high levels of birds of prey, around 200 nanograms per gram. And so from the Arctic, PFAS travel long distances from other regions in the Northern Hemisphere. In Australia, a couple of years ago, the white ibis had elevated levels of PFAS as well. And while there's no manufacturing plants in Australia, we still use quite a bit of the firefighting foam. And these areas have contaminated the use of AFFF has contaminated surface waters that the birds live in. Finally, levels in Antarctica are at the lowest, so less than 1 nanogram per mil in the blood is typically found in birds that live in or around the continent. So just like the Arctic region, this is mainly due to oceanic and atmospheric transport from long distances. Okay, so next I'd like to talk about how we are able to detect these compounds and how we detect them at such low concentrations. So it might be difficult to imagine what a nanogram is, and it's really small. So we're talking the weight of a single human cell. So when you see a concentration like 1 nanogram per liter, imagine the weight of a single cell in a jug of water, but then also imagine the cells broken up into 1 million little pieces and is distributed in that jug of water, right? So really small. Analytical chemistry is the field dedicated to measuring these tiny concentrations of substances. As an analytical chemist, the most important thing to do before you begin any project is select the correct tool for the job. The two most important factors in analytical chemistry are generally the volume of sample and the level of detection needed, right? So first of all, using less sample means that it usually costs less and takes less time to extract the contaminants from the matrix. Because this is one of the because one of the biggest costs is purchasing expensive mass labeled standards. And now you don't always need to use mass labeled internal standards, but that's pretty much the gold standard in analytical chemistry and measurement. And also the less sample you have means you can use less internal standard, right, to keep the cost down. In terms of the level of detection that you require, well, instruments that are really sensitive to low levels are more expensive, but it means that you can detect concentrations down to that nanogram per liter concentration that we're aiming for. Again, you don't always need the most expensive instrument to do research. Many low-cost instruments are still suitable for high concentrations, like if you're doing toxicological studies, right, with concentrations well above what you'd find in the environment. Our lab and all of my work sits up in this top corner here. So I've helped develop some of the most advanced sample preparation methodologies to make sure we can see even the smallest level of contamination. And while expensive, yes, we have access to some of the most advanced instrumentation available, which really allows us to see what other labs might not be able to. All this results in really high impact studies and discovering new things that we've never seen before. So here's a summary of the sample preparation methodologies that I've used over each of the projects. I'm going to summarize them all here rather than doing it individually. They're not the typical methodologies that you might see from other labs, but we've optimized these for cost and time without sacrificing that limit of detection, right? So for example, we've reduced the volume of surface water samples to 50 mils, while keeping detection limits below 1 nanogram per litre. One of my projects was actually validating a novel extraction technique for PFAS in blood and serum. So we wanted to use as little blood as possible so we don't cause too much harm or distress to the animals. And we just want to use a fraction of a milliliter for the analysis. So what we do is we run the blood and serum through a new kind of sorbent called enhanced matrix removal, or EMR. And this removes all of the phospholipids and some of the fatty acids from the sample, leaving behind only our target chemicals, in this case, PFAS. So using this method, we can measure concentrations in serum down to 0.1 nanograms per mil, which is around 10 times lower than what you typically find in your blood right now. We actually evaluated the performance of EMR against some other really common methods used by other labs. And one of the first things we found is that long chain PFAS, which are indicated on the x-axis, are they're not extracted very well with typical HLB or wax methodologies to clean up the sample. What we think is happening here is that the PFAS are sorbing strongly to the proteins and phospholipids in the blood, and that solvent isn't strong enough to elute the PFAS from it. On the other hand, EMR, which is in the top left corner, we use a stronger solvent, acetonitrile, to really rip the PFAS off the proteins and make sure they go into solution. And it worked really well. So we tested the recovery of over 50 PFAS at a low concentration of 5 nanograms per mil. And the EMR cartridges performed the best overall. And keep in mind, this is only working with 200 microliters of sample, just a few drops of blood. Finally, I wanted to talk about how the method, how I helped them develop a method for extracting PFAS from solids, like liver, bird livers, for example, or even soil. And I think we have time for a little live demonstration. So let me just get that set up. And instead of showing you a slide, I can actually do it in front of you here. Okay. Okay, so those at home, they have to look on a little screen, but that's okay. All right, so what I've got here is, here we go. So I've got 5 mils of acetonitrile, I've got 5 mils of water, and in here I've got about 1 gram of sample. And so, safety first. All right, so I'm just simply going to add both the water and acetonitrile into the 1 gram of soil. So what's going to happen is the acetonitrile is going to mix with the water and the soil and start to rip off the PFAS that are sorbed in the solids. and start to bring them into solution. What I'm going to do is give this a quick shake, mix everything up. In reality, what I do is actually put this on a vortexer for about half an hour, and then also put it in a sonicator for about half an hour. But what you can end up seeing is you have a really homogeneous liquid of water, acetonitrile, and soil. So some of the tannins in the soil have gone into solution, and it's become really dark and gross. The next step is we're going to add about a spoon of salt. What this salt is going to do, it's going to react to the water, firstly, but it's also going to make the water more polar as well. And so as a result of that, the water and acetonitrile will actually separate into two distinct layers. Oops. That went everywhere, but it's okay. All right, so what's going to happen is there's going to be an exothermic reaction as the salt reacts with the water, and we should start to see a bit of a separation here. All right. Yeah, that seems to be working. So let me see if I can get this on camera here. So what you can see here is we have a clear solution on top, which is the acetonitrile layer. And then everything underneath that is the polar water that's grabbed all the other contaminants that we're not interested in. And when you put this in a centrifuge, it'll actually separate into like two clear layers. But the next step is we're going to take a little bit of this and clean it up a little bit more, because really it's not really good enough to put directly onto the instrument just yet. So here I've got a little vial, and this has actually got some C18 and PSA. And this is going to take any of the other really long chain fatty acids or anything that made it into the upper layer. So I don't need much. Just want to take that top layer of acetonitrile off without getting any of the water layer. Okay, put the lid on that. So make sure we can actually see this a little bit better. I did bring a centrifuge. Okay, so I'm going to chuck this in the centrifuge. This doesn't take very long. All we want to do is really get all that sorbent that was in there and start separating it out. Okay, and now I hope you can see that we have a really clear solution. And this is now ready to inject directly onto the mass spectrometer. Okay, so that's the little demonstration there. This is called PFECHS, which is quick, easy, cheap, robust, and safe, which is why I could do it all here in front of you right now. Okay, I think we're back. Can see everybody. Okay, see me online, hear me okay? All right. So that's a little live demonstration. Now we have our sample of PFAS extracted into a nice clean solvent. We can use the liquid chromatography mass spectrometry to measure the concentration of PFAS in this sample that we just made. So I've selected this particular tool for all my projects, and this is a culmination of decades in LC-MS research. And so now we can detect chemicals with more sensitivity and accuracy than most other mass specs on the market. It's great. Very simply, the liquid chromatography all happens on the left-hand side of the instrument. And this is how we can separate the compounds based on their solubility. We actually change the concentration of the mobile phase very gradually so that all the compounds in the mixture come out one by one. That's what you can see here, that x-axis is actually time. We can actually control it so strictly that when we do it hundreds and hundreds of times over and over again, it always comes out at the exact same time. Then the liquid is directed into this box on the right, which is called the mass spectrometer. And this instrument is tuned to the precise mass of each of the compounds. And when they hit the detector at the back, we register that as a peak like this. In fact, we actually measure 2 mass fragments simultaneously from the compound, and the ratio between the 2 fragments is always the same. So this is the precise transition ratio of PFAS that we've measured. So all of the things I've mentioned so far help us to confirm the chemical that we're measuring is actually the chemical we think it is. So retention time is the first key. If it elutes at the same time, well, that's a pretty good indicator that it has identical chemistry to the target that we're looking at. Then we also look at a few other things like signal to noise, the calibration range, and the level of the internal standard. One of the best ways to make sure that what we're measuring is what we think it is by using that transition ratio or the qualifier ratio. This becomes super important when you have isomers. So an isomer is 2 different compounds with the exact same mass and similar retention times. So here are 2 PFAS. Both have the exact same number of carbon, fluorine, oxygen, and sulfur atoms. So they have an identical mass to each other, although you can see they're 2 very different chemicals structurally. However, when we look at the transition ratio from a sample, we find that the sample, which is on the top, matches the known standard of PFECHS, which is at the bottom. And so the graph in the middle is actually just both those lines mirror imaged on each other, so you can see them more easily. So we have a high degree of confidence that this compound is actually PFECHS rather than PFOS or any other chemical from that matter. Simple, right? Yeah, well, keep in mind that this is how I've carefully selected and optimized for the tool of my trade. It's nothing more than that. It just provides me with a concentration. So I've simplified this process for your own sake, trust me. But you can ask me any questions about the analytical method later, or they'll all be published in the literature at a later date. So the next part is interpreting what these numbers actually mean. I'm going to show you some real examples of how we use data from this instrument and interpret that. And so the first thing I'm going to talk about is the wastewater treatment plants. So on the left here, you can see the total concentrations of PFAS from 19 different wastewater treatment plants all across Australia. So each column is the sum concentration of around 17 individual chemicals. But the individual concentrations aren't important here. What I want you to look at is how variable the concentrations are from treatment plant to treatment plant, right? Some plants have around 10 to 20 nanograms per litre, while others have over 400. Not only that, but have a look at the change in concentration for an individual PFAS as it goes from the influent to the effluent or the discharge. So these compounds on the top row are PFCAs, and they're increasing in concentration from the influent to the effluent. That means these compounds are somehow being enriched, or more likely that there are precursors in the influent that are oxidizing to the terminal products that we're now seeing at the end. In fact, all the evidence points to the precursor degradation, which happens in the aerobic and anaerobic digesters that the water is treated to. So we already know a couple of years ago that concentrations of PFAS will change between plants and different areas, and it will change over the course of the treatment in the plant as well. But we've never really understood how these concentrations change over time. Are these concentrations representative of the whole day or the whole week or the whole year? So here are some results from a 10-year study of PFAS concentrations at a wastewater treatment plant from my colleagues up at the University of Queensland. And what they found is the concentration of many PFASs had reduced in overall concentration between the years of 2010 to 2020. That's great. So we hope to see further decreases in PFASs emitted to and discharged from treatment plants in the future. It also answers our question if concentrations change from year to year, and we can clearly see that they do. But still, there's a question that remains. Do they change from day to day? Well, there isn't any data available to answer that question on whether inter and intra-day variations of PFAS in wastewater treatment plants. So here's the method for our experiment. We sampled the influent from a treatment plant every hour or so over 7 days straight. And we analysed each sample individually, 168 samples in total. So with the treatment plant, there's a maciator and a pump that comes from the primary influent pipe. It's pumped to an above ground manifold where the samples are taken every hour into their own little sample container. So this is all automatic. Now we don't expect there to be any storm water to be coming through the network because we sampled at a time that had low precipitation. Actually, there was less than 1 millimeter across the whole catchment area for that entire week. So we're not really worried about that. But sometimes you do see ingress of stormwater and groundwater to the sewer networks. Also, this was done before COVID restrictions were in place, and it was done on a normal work week without any public holidays or anything else that might affect our results. What we found is really interesting. For a number of reasons. So first of all, you can see straight away that the total PFAS concentrations per day change quite a bit. So we've got Monday, Tuesday, Wednesday, Thursday, Friday, Saturday, Sunday. And what this means is that if you took a sample from the treatment plant on Monday, you might not be taking a representative sample of concentrations for the entire week, right? Now we only have data for 1 week, but as you can imagine, this might also change from week to week as the time goes on. But at the end of the day, this is going to be really useful for people who are conducting further research at treatment plants because you really need to think carefully about how you're sampling to avoid this kind of bias, right? Point number 2 is these concentrations of FTSAs are really elevated due to the presence of one compound in particular, and it's called 6,2-FTAB. And this is commonly used in newer formulations of fluorotelomere-based AFFF formulations that were developed when PFOS was being phased out. So it's a really complex molecule that was designed to break down easily into short chain PFCAs, which don't tend to accumulate in humans or wildlife very strongly. So we're seeing some relatively consistent discharge of these AFFFs to the treatment plant from somewhere. The third point is the concentration of DIPAPs. DIPAPs were detected in wastewater for the first time in Australia. So these compounds have been detected before in sludge and biosolids. However, their presence in water was a little bit unexpected because they sorbed so strongly to the sediment fraction. And we know from previous research by Damien Moody in 2020 that concentrations of DIPAP are really high in biosolids. So we must be seeing some of that equilibrium of absorption here into the water. And finally, we see really elevated levels of PFSAs on Friday and Saturday. So these are due to pulse events of PFOS. You can see 2 really clear events, one on Friday afternoon at about 4 p.m. and a second event early on Saturday morning at about 5 a.m. Again, it's only speculation as to the source of these pulse events, but high concentrations like this are usually associated with trade waste inputs. So we suspect it could be like an end of week washdown at some factory or multiple factories. And that delay in time on the Saturday morning could be because it's traveling through the sewer network to the treatment plant. Yeah. So lots to digest here, but I want to take you back to that question of when is the right time to sample. What if you took a sample at 4 p.m. on Friday? during one of these pulse events. There's no way to really know that you're sampling during a pulse event. Is that really representative of the average concentrations? Not really. And this is true for other compounds, not just PFAS. So here are the individual concentrations of 15 PFAS that were detected in the majority of samples over this week. The blue bars are the average concentrations and the range if you sampled during business hours, so that's 9 to 5, Monday to Friday. And the orange bars are the average and range of concentrations for after hours, so including the weekends. And there's a significant difference in the average concentrations for about 10 out of 15 of these compounds. So some of these compounds had the greatest, these 5 had the greatest effect size, which means the difference in the average concentration was the greatest. And we're looking at the C6 to C9 PFCAs, which all had greater concentrations during the day, and one DIPAP, which actually had greater concentrations at night. So what we see sometimes in the literature is people using grab samples taken during business hours, and then using that information to estimate the total mass of PFAS that are, being discharged from the treatment plant over the day or week or year. And what we found is that you can't really do that. You need to take pooled 24-hour samples or longer. Now, I'm not saying that grab samples are bad, but it does depend on how you're interpreting that data. And essentially, you can't take a sample from a single hour or even a single day and use that to impute concentrations over time. And it very well may be a similar case in surface waters or fresh waters from the environment too. So it's something good to keep in mind whenever you take samples from any water source. Okay, all right, so we're almost there. This is the last project that I want to share some results with you. So taking all that knowledge, let's have a look at levels of PFAS from an urban lake in Melbourne. All right, so Albert Park is a lake located just south of the CBD in Melbourne. And given the history of the area, there's no evidence to suggest that major contamination of legacy PFAS has existed, despite being used as a municipal dump in the 50s. Well, we know PFAS weren't even being manufactured, well, around the same time. Then in the later half of the century, when it was occupied by the Australian Defence Force, it was only a barracks, and there wasn't any history of aviation usage in the area that's usually associated with Defence Force contamination. However, the Albert Park circuit was opened in 1996 for the Formula One and other high performance motorsport racing, although this isn't really a known source of PFAS contamination as far as we know. Then in 2008, a stormwater diversion project was completed to make sure that the volume of water in the lake stayed really constant at a high level. And this will discharge something like 300 megalitres of water per day, diverted from the stormwater in the surrounding suburbs. And we found that this water and sediment in the lake had really elevated concentrations of PFAS compared to other nearby surface waters in Melbourne, right? In fact, anyone looking at this with some knowledge on typical surface water concentrations might be a little bit surprised about what we found here. I mean, we were. The most abundant compounds were PFOA, which had an average concentration of 47 nanograms per litre, PFOS at 31 nanograms per litre, and PFECHS at 12 nanograms per litre. But at one site, 150 nanograms of PFOS per litre was detected, which exceeds Australian guideline values for 95% species conservation. And these concentrations are consistent with surface waters impacted by wastewater, industrial or agricultural inputs, not really from residential areas. So these increased levels of PFAS in the water and sediment resulted in the contamination of the population of black swans that live here, and also in roughly the same proportions. So swans just eat plants and algae from around the lake, but these levels of PFAS are more in line with levels you typically see from species in a higher trophic level or from a really contaminated site, like those seabirds or eagles that we saw earlier that eat contaminated fish. I think this is a product of the highly impacted surface water and that the birds are feeding from. And concentrations of some PFASs like long-chain PFCAs were found around the same level of birds that were next to manufacturing facilities. Now, again, it's hard to say if these birds are experiencing any negative impacts due to their exposure. This is just a single study, so we definitely need more research to follow up on these findings. And one last thing, we did find the novel cyclic PFECHS in water and blood samples from black swans at average concentrations of 27 nanograms per mil or litre in water. So this is in the same range as waters found adjacent to airports in North America and Asia. So overall, the concentrations this high and the presence of PFECHS were really unexpected, and it's been accumulating in the swans. So we were able to determine the bioaccumulation factor for PFECHS in birds for the first time, which was around 2.8 log units. So the bioaccumulation was a little bit lower than something compared to PFAS, which has a bioaccumulation factor of about 3 log units. And this will really impact it will affect how we assess the risk of PFECHS to birds in the environment from this study. And also I wanted to show you the relationship that we observed between concentrations of PFAS in serum on the x-axis and in their excrement on the y-axis. And there's a really strong correlation here between these 4 compounds, as well as the sum concentrations of all the PFAS together. So this is really cool and might allow future researchers to only take excrement samples to monitor the PFAS concentrations in their blood over time, right? And this is great because it's a novel, non-invasive sampling method for kind of looking at the body burdens of PFAS in birds, at least for this population of swans. And of course, there are some errors in the estimates of the PFAS levels in the blood compared with the excrement. But hopefully with some follow-up studies, we can really start to focus this model down and really look to get it more accurate. And I just want to take a step back again and look at the detection of PFECHS at Albert Park. So there have been some scattered reports of this compound in the Northern Hemisphere near airports, like I said. You can see that the highest concentrations were reported downstream from an airport in Beijing, and the lowest concentrations were from an ice cap in Canada. And so the presence of PFECHS in Canada was mostly associated with long-range transport. So it's also possible for this compound to move long distances in the environment. And since Albert Park isn't near an airport, and it doesn't receive any discharge from industrial areas, the only explanation for the source of PFECHS to this system that we think could be the use of aviation-grade hydraulic fluid for the high-performance vehicles at the Albert Park circuit. So we're proposing that these vehicles may be using the PFECHS in the hydraulic fluid, and this could result in the contamination of this compound to the environment. There could also be AFFF exposure from the site. However, PFECHS isn't really associated with any AFFF that we know yet. The source of PFAS to Albert Park is something that Brad Clark's group will definitely follow up with in the future. But if our suspicions are true, and it's coming from high performance vehicles, then this will also have international implications. And sometimes with human ecological health impacts. So there's similar circuits like this around the world. Some of them are in developed and densely populated areas. And what if an area around there was using this water, surface water or groundwater for drinking water? You could lead to significant human and ecological exposure. Okay, almost there. I just want to wrap this up with a few final thoughts of what I've learned over the last couple of years. So yeah, I definitely recommend that investigations into PFAS exposure for birds continues in Australia. Even with the work that I've contributed, there's still a huge knowledge gap in many parts of the country. And this extends really to the whole Southern Hemisphere. There's a real lack of literature there. Even though PFAS manufacture is primarily done in the Northern Hemisphere, nearly every country on the world will have humans and wildlife exposed to AFFF and long-range transport. So we definitely need more investigation to known sources like wastewater treatment plants and defense sites. And sometimes these areas are really important habitats for wildlife. And in order to mitigate the effects of these compounds, we need to understand more about the extent of contamination in Australia. And birds are a really unique case study here because some states will allow large-scale hunting programs. So here in Victoria, the EPA posts this advising hunters not to consume waterfowl from an area that was heavily impacted by AFFF exposure. And so the PFAS levels detected in birds from this area exceeded all the government guidelines for what's safe for human consumption. The complete toxicological impacts of PFAS is also lacking. There have been some studies to suggest that there are non-lethal impacts of PFAS that might affect things like growth and reproduction, and we don't really understand that completely just yet. And finally, to bring all this research into focus on a global scale, many of you will be familiar with the concept of planetary boundaries. And PFAS would fall into the category of novel entities, where there's really limited information on the thresholds that could potentially cause irreversible harm to the planet. And I agree with the author's recommendations here that we must learn from our past mistakes, and importantly, we must use precautionary and preventative actions to address these issues. So here's the ALEC Class of 2021, and yeah, I'd just like to thank everyone in the team for all their help. It's been really fun last couple of years, and I really couldn't have done it without them, especially Brad Clark, who I've been working with for the last 5 years. Yeah, I wouldn't be standing here today without Brad. So thank you very much. And there's a whole bunch of other people that I've worked with over the last couple of years, from RMIT, Agilent, Melbourne Water, Mark Green's lab, the Water Research Australia, University of Tasmania, and Melbourne Water. And I'd like to thank you all too and thank you all for coming. And finally, my parents, Steve and Sarah, thanks for coming down from Sydney today, and it's good to see you. And last but not least, Katie, my beautiful partner, who's put up with me for the last 4 years and has been always really supportive. And yeah, I couldn't have done it without you. So thank you.